Hi, my name is Shithuzen, and welcome to my video on how to stop Renfro from buying any more wigs. Let's begin with the basics. In order to save Renfro, we need a protein. We'll call this Renfronin. In order to make a protein, we need a messenger RNA. In order to get a messenger RNA, we would need DNA, and this DNA would be located in the nucleus. This is going to be the DNA that will change Mr. Renfro's life forever. Well, no guarantee. As you can see in this image, there are introns and exons. You probably also notice that there are pairs of threes. These are called codons. You know, the ones with the letters on them? Together they make up the strand of DNA, both introns and exons. Now, in order to make the protein, we need a messenger RNA, like I said before, which is also known as mRNA, M standing for messenger. In order to make this mRNA, we need RNA. This is accomplished through transcription. The thing that allows for transcription is the top box, which is the thing on the left, or called 3'. The purple blob on the bottom is the transcription factor. The red box, blue box, and green box are the RNA polymerase. Their job is to match the template with the matching RNA codons. The template is the bottom half of the DNA. The reason why the top portion will look different this time is because A's are matched with U's instead of T's. Besides for that, the next top portion should look almost identical to the previous top portion. Once the entire template is scanned and the codons are paired, then the RNA polymerase will meet the stop codon at the right side, or 5'. This will form the RNA polymerase to stop, as you can imagine. By the way, this is all occurring in the nucleus. Now that we have our pre-mRNA, we need to filter it. In other words, we need to remove the introns. This is accomplished by proteins called spliceosomes. It's the blue ball with black lines. Those black lines are snRNA. The entire ball is snRNP. The P at the end of the name represents protein. If you're wondering, snRNP stands for small nuclear ribonucleoproteins, because biologists have an irrational fear of small words. Now, we need multiple of these proteins to splice or cut out the introns. I also have simplified the pre-mRNA so it will be easier to see and also easier to work with. So, the spliceosome will attach the introns, cutting them out or curling them into a round formation. The round formation will be taken away and maybe reused. We still don't know for certain why there are introns, but they do exist. By the way, this is still occurring in the nucleus. This is an image comparing the size difference between the pre-mRNA and post-mRNA. As you can see, it is smaller. Also, notice the 5' and 3'. They are now cap and poly A tail. So this fully processed mRNA can now enter the cytoplasm to make a protein. Now, in order to make a protein, we need a ribosome. Before we can use a ribosome, we need tRNA and an amino acid. Remember, proteins are chains of amino acids. The only problem is that the amino acid can't travel far on its own. That's where the tRNA comes in. It stands for transfer RNA, and as you can imagine, it transfers things. In order for it to carry the amino acid, it needs an enzyme to help attach it. This process of pairing and attaching is called amino acyl tRNA synthase. In order for the tRNA to connect to the mRNA, it needs matching codons called anticodons. In order for the amino acid to become a chain, we need a ribosome. In the ribosome, there is an A site, P site, E site. The A site introduces the tRNA amino acid complex to the ribosome. The P site detaches the amino acid carried by the tRNA. The E site throws away the tRNA so it can be used for later uses. So as the ribosome moves down the mRNA, tRNA carrying amino acids enter the A site, move to the P site, and exit through the E site until a release factor enters the P site. The release factor tells the ribosome to disassemble. This leaves the protein, also known as renfronin, behind.